In our previous videos, we looked at how to identify the epithelial tissues. We said that to do so, we find cells that are packed closely together. We follow that up by asking, do the nuclei appear to line up roughly in straight rows? And if yes, we're looking at some kind of epithelial tissue. But what happens if we're looking at a tissue that has cells that do touch and pack closely together, but their nuclei do not line up in relatively straight rows? In that case, we might be looking at our fourth category of tissues, that is, the muscular tissues. Once we think we might be looking at a muscular tissue, we have a couple of special follow-up questions that we only ask for the muscular tissues. The first one is, do we see striations? That's a sort of wavy kind of appearance, or tiger stripes, a pattern of dark band, light band, dark band that goes all the way across the tissue. Two types of muscular tissue have striations, the skeletal muscle and cardiac muscle. One type does not, the smooth muscle. Well, if two of my muscles do have striations, then I need something that tells me the difference between them. So my second follow-up question for the muscular tissues is, are the cells straight or not? Let's take a look at skeletal muscle to work our way through our series of steps. We always start by finding cells, and we typically do that by looking for dark staining nuclei. Here, I've highlighted a number of them. Once I have those nuclei, I look around them to try to find the outline of the plasma membrane. When I do that here, I find that all of these nuclei belong to one supermassive cell. I'm looking at a very long, multinucleated cell. There's only one type of tissue in the body that fits that description. That's skeletal muscle. But let's look for some of the other characteristics. I need to find some cells, so I'm going to highlight a few. Now that we have those cells, we ask, do the cells appear to pack closely together? And they do. There's not a lot of non-cellular stuff in this image. Then we ask, do the nuclei in the cells appear to line up in rows? Well, in this case they really don't, because the ends of this cell would attach to the origin and the insertion for the muscle. That means that one cell goes the entire length of the skeletal muscle, so the cells don't line up in a row because each cell goes the entire length of the muscle. Now that I've decided that the cells don't line up in rows, I might be looking at some kind of muscle. So I ask my first special muscle question, which is, do I see striations? And if I look closely at this image, I can see what appears to be sort of tiger stripes going across each of these muscles. Now, that tells me I'm looking at either skeletal or cardiac muscle. Now I follow that up with my second muscle question. Are these cells straight? And yes, they are. Striated, multinucleated, straight cells tell me that this is skeletal muscle. Let's contrast that with another kind of striated muscle, that is, cardiac muscle. I follow the same series of steps. First, I need to find cells, so I'm going to highlight several cells here. I could find those cells because of a special feature for this type of tissue that we'll name here in a little bit. And what I find are cells that do touch each other, so they are packed pretty close together, but the cells don't line up in a nice neat row. Instead, the cells kind of branch and go off in different directions. So I might be looking at some kind of muscle. Because I think it's muscle, I ask, do I see striations? And here I can see tiger stripes going across these cells. They're not as dark or as defined as what we saw for skeletal muscle, but they are there. The other thing that's special is I can see the ends of these cells. I know where one ends and one begins. I couldn't see the beginning and the ending of a skeletal muscle cell because they were so long. So I must have a different kind of tissue. I can tell where these cells end and where they begin because of a feature that's unique to cardiac muscle. These very, very dark bands that are darker than any striation for the muscle here is a special feature called an intercalated disc that plays a really important role in coordinating the contraction of cardiac muscles. Intercalated discs are either going to show up as a very dark band, as they do in this image, or as a very bright white band going across the cells if we use a different kind of stain. Either way, it's going to be a distinct band that is either darker or lighter than the striations on either side. So here I have cells that touch but don't necessarily line up, so it's a muscle. I see striations. I see the beginning and the ending of the cells because of the intercalated discs, and I see that these cells are a kind of branched shape. All of those characteristics tell me that this is cardiac muscle. The third kind of muscle, and the last tissue we need to name, is called smooth muscle. And smooth muscle is called smooth because it lacks striations. It doesn't have that wavy type of appearance to it. But we still need to go through all of our steps. Step 1, I need to find some cells. And here, I find these cells that are sort of a fusiform shape. That means that they're pointed on both ends. Now that I found some cells, do they pack closely together? Yes, they do. Do the nuclei line up in nice, neat rows? Not really, so I might be looking at some kind of muscle. Because I think I'm looking at muscle, I ask, do I see striations? And no, I don't. So this is smooth muscle. Now, I want to make sure that you're very careful because it's very easy when we're first naming tissues to confuse smooth muscle with the stratified squamous kinds of epithelia. So it would be a good idea to take a look at several examples of each and see if you can find a difference. One thing that you might find that's different is in some images of smooth muscle, the cells are kind of separated by a small white space that allows us to see that they're not all packed directly on top of each other the way epithelial tissues are. So please make sure you look at several examples of smooth muscle and epithelial tissues to try to find those differences. Coming up, we need to take a look at how we put this entire process of our decision tree together.